Hello, my name is Sean Calingayan, and I will be your host for this two-part lecture series on forensic proteomics. Please note that this lecture series serves as an introduction to the world of proteomics and may not cover in-depth details regarding the discipline. Thank you for watching. This lecture series will cover the general overview of proteomics, how you would conduct a proteomic analysis, applications to forensic science, any challenges and logistics, and finally end off in a case study. To start off, let's go over the definition and what proteomics is. So proteomics is defined as the study of the overall protein portfolio within a cell or tissue sample, including adaptations with changing environmental conditions. This essentially means that within a sample or a cell, you are analyzing all the proteins that are present and what concentrations they might be in as a result of any changing environments or conditions that the cell might be placed in. And as this is a forensics lecture, let's remind ourselves what the definition of forensics is. I'll give you a minute. Hopefully you got it. It is the application of physical or natural sciences to a court of law. So overall, forensic proteomics, we are taking any essentially instrumentation, any methods from proteomics, and we're applying it to criminal investigations, perhaps civil, or maybe even agricultural or environmental cases. So before we look at how proteomics can be applied to forensics, let's first look at its original usage in biological and biomedical sciences. So proteomics can be used in biological and biomedical sciences to determine disease pathways, biological regulation mechanisms, and characterize microbial communities. An example of this is how researchers use proteomics to determine the mechanism for SIDS, or sudden infant death syndrome. So the protein on the right is 1433. This is a protein that controls serotonin levels within the body, mainly the cerebrospinal fluid. Researchers found that decreased levels of this protein and tryptophan hydroxylase indicate that an infant is developing sudden infant death syndrome, unfortunately. How they do this is using analytical instrumentation such as liquid chromatography and tandem mass spectrometry. And this is the schematic for an instrumentation setup for liquid chromatography and tandem mass spec. The usage of analytical instrumentation like tandem mass spectrometry or tandem mass spec allows for a lot of advantages for proteomics. There are three main points that I want to go over regarding advantages. The first is sensitivity and selectivity. You can apply this to actually any type of sample, and you really don't need that much sample preparation, mostly just protein purification. And additionally, for more sensitive or more selective, you could adjust the instrument to manipulate any peptide ions to increase the accuracy, precision, and sensitivity. The second advantage is comprehensive analysis. You can get both qualitative and quantitative data. So essentially, is the protein there? And if so, how much of it is in the sample? And finally, multiplexity, which is very important and very, convenient. Multiplexity just allows for high throughput analysis. So you can do multiple at a time, and this is because mass spec allows for concurrent analysis. So before we move on to the workflow and how to conduct proteomic analysis, let's just take a minute and think, why proteins? The answer for biological and biomedical sciences is more intuitive than it is for forensics, because recall the definition, you're looking at proteins and the difference in expression and changes for changing environments, which explains, disease pathways, biological mechanisms, stuff like that. But forensics, we already have the gold standard. We have DNA, right? And we have other forms of biological evidence, such as hair and, again, DNA, fingerprints, blood. So why? Why are we looking at proteins? Well, typically, proteomics and proteins in general are employed when DNA is too degraded or potentially contaminated, which interferes with the proper DNA analysis. And As you can see in the central dogma on the screen, we have DNA, which then leads to mRNA, which then finally leads to proteins. But if you're paying attention, you might see, well, why not use mRNA, right? It's the closest thing to DNA. It's only 1 degree away. But if you have any knowledge of molecular biology, we would know that mRNA is unstable. And the only job it has is to take the DNA and the information from the DNA and give it to the ribosome to make the protein. And while proteins aren't exactly, the most stable, well, they are, but in certain conditions, it is more stable than mRNA. And they do essentially last longer and have a longer half life than mRNA, which is why we typically use proteins or proteomics and proteins, again, as a secondhand method when DNA is unavailable to use. So, moving on to the workflow and how to conduct proteomic analysis, we have essentially 4 steps that I like to break it down into. So, number one, we have sample preparation, where you're purifying your protein and maybe digesting it too, which we'll talk about a little bit later. Number two, the separation. So, we're using liquid chromatography to separate a singular protein from a bunch of proteins, essentially picking out which protein that we want. Third, we're using tandem mass spec, or MSMS, which is the notation used in writing, to see how much protein is there. Is my protein there? Essentially, this is our detection phase. And finally, data analysis. How do we essentially recognize our protein within the data and how we analyze it, essentially? So the first step in proteomic analysis is essentially getting your protein and preparing your sample. So your sample could be any of the 5 things I have on the screen, even more. There are more sample types that are available. Anything with a protein in it can be subjected to proteomic analysis. So the things I have on the screen: tissue, insects, powders, liquid matrices, and non-animal cells. And depending on the nature of the sample, so any of the 5 and then some, they will be subjected to different types of preparation. So first we have homogenization on the left in the red, which is literally physically grinding your sample to extract your protein. So this can be done using cryogenic grinding, so freezing it and then smashing it to get your protein out. You can use a mortar and pestle just straight up, and then I think they call it a tissue tearer, which is essentially just a blender. But yeah, so any physical grinding and physical laborious activity to get a protein out is the homogenization. More delicate is chemical lysis. So this is when you're using water or buffer to essentially explode the cells to get your proteins out. And finally, dissolving in buffer and water, which is the most intuitive. You're just literally adding buffer and water to your sample. And so based on those descriptions that I've given you about each sample step or sample preparation step, I'll challenge you all to, sort of place each sample type that I have on screen into where you think it should go, right? So these 5: tissue, insects, powders, liquid matrices, and non-animal cells, sort them into, how you prepare them, essentially. And the answers are here. Essentially, this slide is the answer key. So you have tissue and insects under homogenization, so you're physically grinding those. Chemical lysis, you're kind of only using that for non-animal cells, so bacteria or plant cells. And finally, dissolving in buffer/water, you have powders and liquid matrices, mainly because you're just trying to get these into a form that can be subjected to liquid chromatography, if that makes sense. So, we have our protein. Now what? Well, we have 2 things. The question remains: to digest it or not to digest it. If we choose to digest it, we call that the bottom-up method. And this reduces the analyte size from whole proteins to multiple polypeptide chains. You're using the enzyme trypsin, which cuts at lysine or arginine residues, and this creates predictable digestion products. We mainly use trypsin because if we use a different enzyme to essentially cut the peptide chain, we might get too long of a chain or too short of a chain for proper analysis. Now, we have something called the protein interference problem. Let's think about it for a minute. If you have a bunch of proteins in the sample and you chop it all up, if you chop all of them up, you might have similar chains, right? And so this is one of the issues with the bottom up method, is that we encounter the fact that some peptide chains can be found in different proteins in the same sample. We'll talk about how to work around and any solutions to the protein interference problem later, but right now just keep that in the back of your head. Conversely, after the bottom up method, we have the top down method. Way simpler. Just leave it alone. Leave the sample alone. You're doing the proteins whole. You're just going to put them through liquid chromatography and the instrumentation just alone, like whole. And this provides more qualitative data rather than quantitative. This is essentially just like, is my protein there or not? The top down method is typically more used for identifying post-translational modifications on proteins. So that's why it might not be as relevant in a forensic sense. So we have our first of two instrumentation steps right now. In proteomic analysis, this first step is the separation step, where you're employing liquid chromatography. There's nothing much to say about this liquid chromatography. It is reverse phase, stationary phase, so your hydrophobic stationary phase and your aqueous mobile phase with the increasing organic solvent as you move on. It really is just basic liquid chromatography. If you have no idea what I'm talking about, I would recommend a small little Google search. It is pretty simple. You're essentially just, separating your analyte into small parts and then picking out which of the parts is the one that you want. That's basically what chromatography is. But yeah, pretty standard. Moving on to the second instrumentation step, we have mass spec. So recall that I said it is tandem mass spec. We're just going to cover one mass spec and then we're going to move on to what tandem mass spec means. So mass spec, or a mass spectrometer, essentially has a varying ion source and at least one mass analyzer with ion optics and collision cells that are optional. You have various ionization methods. There are many out there, but these are the two most common for proteomics. Electrospray ionization, where you're spraying the solution through a small needle-like emitter under high voltage to give that sample a charge, which then creates the ions. Or you have MALDI, or matrix-assisted laser desorption ionization. The mechanism is there at the bottom, where you mix the sample with a certain matrix, you dry it on a steel plate, and then you irradiate the dried sample with a laser. One thing to note, I did mention that these two are very commonly used in proteomics. This is because these are soft ionization methods, and so these methods don't create really, or this prevents severe fractionation and severe breaking of the molecules in the sample. And another thing to note is that MALDI, so the bottom one, is an independent or ambient technique. So this is able to be in situ rather than like attached to the instrument itself, if that makes sense. You're able to essentially more easily bring it anywhere. It's more accessible, that's the word. It's more accessible, the MALDI ionization. Okay, so we covered the ionization sources, the soft ionization from the either ESI or MALDI. So now we gotta analyze the mass of the ions. So the mass analyzers use an electric or magnetic field to separate ions based on physical properties. Uh, this is essentially a detector. You have hybrid detectors such as triple quadruple, ion trap, orbit trap, everything listed there. These are capable, hybrid detectors are capable of multiple rounds of measurements, selection, and fragmentation. And they are essentially the source of the resolution. So the type of detector you're using or the mass analyzer essentially determines how accurate or how precise your measurements are going to be. And so just a note there, narrower peaks from your mass spectra is equal to more precise and accurate mass to charge value. And at the very bottom, we have low resolution detectors and then high resolution detectors. So what we just covered was the instrumentation for a single mass spectrometer. I want you to take that and double it because that's what tandem mass spectrometry is, essentially. You're just doubling up on mass spec, and if that confuses you, I'll just break it down for a generalization where you have your sample that you break into ions, and then you analyze the ions, right? For in the first mass spec. For the second mass spec, we take the ions from the first one, we break them up into smaller ions, which we call transitions, and then we analyze those. Hopefully that should clear it up if you're still confused. And so, two things to note here. Number one is that because we're using ions and not just an overall sample now, we can filter out specific ions that we want to analyze compared to the first mass spec. So that's this first point here where ions of desired mass to charge values are isolated by controlled electric fields. You're essentially picking and choosing. The second point and anything below the second point is essentially how we determine the transitions and how we create the transitions. You're essentially just colliding them with gas molecules, inert gas molecules, so they don't react. And that the physical collision just breaks the bond because of the vibrational energy. That's just physical chemistry stuff. And then the final point is that amino acid sequence can be determined based on the fragments. So we have these very small mass spectra, very specific to an amino acid, or a specific amino acid sequence. And when we put them together, we can get the whole chain of the protein, right? One thing to note for this last point regarding the amino acid sequence, keep in mind that isoleucine and leucine are constitutional isomers. So when you're putting together the sequence, you might have isoleucine or leucine because they are the same mass and they give off the same mass spectrum. That's just one thing to note. And so I recognize that could have been a lot of information regarding instrumentation to process. So here is hopefully a helpful tool in this visualization of what happens during the instrumentation that it could help you just understand it better, right? So here's our sample. The red is our specific analyte we want to pay attention to, which we're going to follow along, right? And I want you to keep in mind, this is just a general, instrumentation workflow. So keep in mind what our analyte or what our starting sample might be if we did the bottom-up method or the top-down method, okay? So I'll just leave these I'll call them analyte and sample, but I'm not going to say if it was a protein or a peptide because that just allows for a little more ambiguity and if you were using the bottom-up or the top-down method, right? So we have our sample. We're going to subject it to liquor chromatography. And these, the black ones, the black circles, are going to separate separately. That's a funny way to say it, but they're essentially going to break apart from our main analyte, which is the red orange circle. And now that's the chromatography done. And now we're going to go through mass spec. So we're going to zoom in onto our analyte. And as you can see from our analyte, we have A, B, and C, which are all protein chain. So this is one interconnected protein chain, which is then going to break apart within our first mass spec, right? So we have a protein or a large peptide chain as our analyte, and then we're just going to break it apart into A, B, and C, which are all protein chains of a shorter size, of a shorter length. And then for our MS2, we're going to look at protein chain A just for this example, but keep in mind this does happen for B and C as well. So for MS2, we're going to break apart our smaller protein chain A into smaller transitions A1, A2, and A3, or AN. So you could break it apart as many times as it physically allows. Just for the sake of this example, we're going to go for 3. And keep in mind, this MS2 will also happen for B. So you will also get B1, B2, B3, and then C1, C2, C3, CN, right? Until, you're physically unable to break it apart anymore, or just, physical chemistry. Just as how there are 2 methods for preparing your sample, recall the bottom up and top down methods, there are 2 methods on how to analyze your sample, the targeted versus untargeted method. To start off, let's look at the targeted method, where you're detecting and quantifying specific proteins. So this essentially means you know what you want, you're assuming it's in the sample, and you're going to set all your conditions specific to that protein or those proteins so that the only data you get is from those proteins, the ones that you know you have and that you want. This is done through single or multiple reaction monitoring, which essentially means that you're either looking at one or multiple peptide ions within the mass spec. That seems complicated. I trust you. Trust me, it's not. This targeted method is favored for protein quantitation because it has a lower limit of detection than the untargeted method. This is because it has less noise. Again, because you're setting your conditions specific to a protein, right? And this typically uses triple quadrupole mass spec. And the jobs or the usages of the 3 quadrupoles are listed below. So the first quadrupole is a filter. You're blocking out essentially everything except your specific protein. And then the second quadrupole, you're breaking them apart using inert gases. And then the third quadrupole is that you're going to take your transitions or your ions and you're going to filter them again to get the specific ones that you want. So that is the targeted method. The untargeted method sounds what it is, right? You're trying to get as much data as possible, which is why they call it shotgun proteomics, because like a shotgun, you're going to just hit as much as possible because you're not essentially just shooting a single bullet. You're doing like the pellets, right? There's no set target protein. You have a higher background signal, so it's less sensitive because, again, you're trying to get as much data, so everything's just being analyzed. The untargeted method uses something called data-dependent acquisition, where you have the peptides are measured first in a precursor scan, and then essentially that informs what you're going to do next. So you start off with untargeted and then essentially goes to targeted. That's essentially what that means, because you're first going to determine what proteins are present or what peptides are present, and then you're going to set conditions specific to that protein or peptide, and then obtain your mass spectra. And that is it for the instrumentation part. The final part of the workflow is the data analysis. So there are different ways to analyze your data depending on what type of analysis you subjected your sample for. So first we're going to cover the 4 methods of untargeted data analysis, so for your mass spectra, and then we're going to cover the targeted mass spectra analysis. So the first method for untargeted data analysis is database searching. And this is essentially just comparing your experimental data to that of a database. Pretty simple. You're gonna score them by a peptide sequence match, essentially just saying how well do these spectra from the database and the ones that I got, how well do they match? And then you could set a threshold to filter out any false positives or any negatives. So essentially, you're determining how much is a positive match, which is a true match, let's say. And database searching can be done via algorithms, so this can be automated, or it typically is done manually. One thing to note is that database searching can only be done if there's a reference genome for the species of origin. Moving on, we have the spectral library search. This is very targeted. So this is a targeted database search. It is very similar, but again, it's not necessarily for or from a database. So the what you're comparing to, your reference, isn't necessarily a database. It can be from artificial peptides, which were synthesized in silico, or essentially just a simulation. It can be a simulated mass spec that you're comparing to. So those are the two essentially quote unquote database searches or comparison methods. The next, these two methods don't necessarily rely on a database. The database is not necessary for them to be successful. The first of these is the de novo identification. You're interpreting it by yourself, essentially manually. So you are determining peptide sequence from the fragment masses and base peptide knowledge. So essentially, you know, or you think you know, you're the mass of the peptide for the base peptide that you broke apart, and you're essentially just trying to piece it together. This can be done out with an algorithm, right, to compile any candidate peptides, but then this has to be checked over by an actual analyst. And the final method is the tag-based identification. The analyst identifies tags, which is a sequence of 2 or 3 amino acids within the mass spectra, and you're going to confirm the sequence based on the prefix and suffix masses, so the masses before and after the tag. Important to note that the suffix masses will vary, or may vary, and which is the reason why this method is not commonly used, but it is ideal for any expected post-translational modifications or amino acid variations. So essentially, what method you'll be typically using is the database or spectral library search, but it is important to note that de novo and sequence tag are also very helpful in certain cases. This chart here is essentially just how any advantages and disadvantages for the 4 untargeted data analysis methods. For the targeted data analysis, I think this is pretty simple. It's one slide. You're setting everything specific to that peptide or protein that you want. So your retention time from your chromatography, your peptide masses, your ion masses, your fragment masses, they should all be very specific to your target peptide. One thing to note here is the second and third point on the slide. A single transition is used for quantitation, and an additional fragment is used for confirmation of peptide identity. So that essentially means that you can use a single transition, so a single small fragment, to identify how much of that protein was in there. But then to confirm the identity of the peptide, you have to use another amino acid or fragment or transition from that same peptide chain to confirm that, hey, this is the same peptide instead of, a separate peptide. And the third point, which I also pointed out, is that if you're developing an assay for a targeted protein, you want to just pay attention to any interfering peptides that might interfere with each other. that sounded redundant, but essentially, some peptides might interfere with the analysis of another. And if that does happen, just pick a different one. Pick a different peptide from the same protein, and you should get the same results, if not better. Okay, the final part of the workflow, important to note, is the protein quantitation. So, how do we get information on how much protein is in the sample from the mass spec? So, you have two types, relative versus absolute quantitation. Untargeted could only result in relative quantitation, whereas targeted can result in relative or absolute quantitation. The difference is absolute can only be achieved if you add internal standards. So how do we determine quantitation or quantity? 3 methods. Number 1, the label-free method, where chromatographic peaks of peptides are compared between samples. Essentially, you're just taking the ratio between one protein versus the other. Simple. Spectral counting. Counting and comparing the number of tandem mass spectra that matches the peptide. Essentially, how many times a certain spectra shows up per sample is equal to the number of protein in the sample. And finally, the heavy isotope method, where proteins are tagged with different isotopes, either the heavy or the light one, and then the ratio of peak areas are calculated for quantitation. This is the internal standard I was talking about earlier for the absolute quantitation. And this is where I'm going to leave off this first part of the lecture. The second part will cover application to forensics and any challenges, and finally a case study regarding forensic proteomics.